Hi guys, welcome to the last video in this lesson on contraction of skeletal muscle, on the physiology of skeletal muscle. I want to talk a little bit about what alters the force of contraction in skeletal muscle. This is the active length tension curve. I'll get to this in a second. This is the active and the passive and the total tension curve, length tension curve. But for the active curve, that is the amount of tension generated by actin and myosin interactions and active contraction, ATP-dependent contraction, you'll notice that as length changes, the force of contraction or the tension generated changes, right? You get a peak here, and this is the optimal length for the skeletal muscle. And in skeletal muscle, your muscle rests or tends to sit at the optimal length. So resting muscle that isn't contracting tends to want to sit at this length where you'll generate maximal contraction if you excite the fiber. And if the length increases or decreases, then the tension decreases either way, okay, from that peak. Notice that this is different from cardiac muscle, which only operates on the ascending part of this curve. So in cardiac muscle, you can never stretch a normal cardiac muscle beyond this peak, and so you never get to the point where length actually decreases tension. The length tension curve for cardiac muscle is just this part of the curve, so that with cardiac muscle, you're always somewhere in here, and if you increase the length, the force of contraction always goes up. That is not true of skeletal muscle. If you go to either side of optimal resting length, you will decrease the force of contraction. So if I decrease the length of this fiber, what happens is I start to lose overlap between actin and myosin. There are a couple things that are happening. If it's really contracted, then what happens is the myosin actually starts to hit the Z line, and you actually get some inhibition of contraction that way. This would be your Z disc here. But beyond that, what happens is your actin starts to run over, your actin filaments on either side run over one another. So if my myosin's down here, my actin's up here, my actin's moving like this. As the muscle is shortened, eventually you end up with this situation where this part of the actin is no longer in contact with the myosin because it overlapped with the actin on the other side. And so you lose myosin-actin interaction there. You lose contact through the myosin has to lose contact with the actin. And so because of that, you get less force generated. At least that's the theory. On the other hand, if we lengthen this fiber, notice what happens is you lengthen it, you've got three sort of like dots of actin here overlapping with myosin. Now it's just two as I lengthen it. So that means there's less force being generated because I have fewer myosin-actin cross bridges. And of course, if I continue to lengthen it, then I lose even more cross bridges and my tension falls, all right? So that's sort of why your active length tension curve is what it is. The active length tension curve simply generated by actin-myosin interactions and through the ATP. Now you'll notice over here on this one, we have extra information. Here's our active curve, it's the dashed line, it's the same as this. And here we have passive tension. And the passive tension is generated not because actin and myosin are interacting with ATP and forming an active contraction. Instead, this is because you're stretching your muscle like a rubber band. So you have proteins and cytoskeleton inside that muscle that really don't want to be stretched beyond a certain point, right? They're like a rubber band. And so if I stretch a rubber band, I generate more tension as I stretch it. If I stretch a cardiac cell, I simply generate more tension just because I'm stretching it, not because I'm stimulating it or causing any active contraction, just because I'm stretching it. And the part of that increase in tension that is just because you're stretching the fiber, that is called a passive increase in tension. So this is the passive length tension curve. And if you add the tension of both of these curves together, the active and the passive, you get the total, where you kind of come to a peak where the active peaks and then you get a bit of a drop, and then you go up again because the passive is starting to become really significant and really high, and so the total starts to rise even though the active tension is continuing to drop, because this passive tension is more than picking up the slack and actually causing the total tension to increase. So this is your total length tension curve here. This is the active part of that, the part just that's ATP dependent and myosin inactive interaction. And this is the passive part that's just the part that is stretching the fiber like a rubber band, okay? All right. This is the force velocity curve. You'll recall this came up with cardiac muscle as well, and it operates pretty much the same way as cardiac. So if I take a muscle fiber and I put a load on it, let's say I put weight on it, the muscle fiber has to generate enough tension to overcome that load in order to contract and shorten. And so the more tension it has to generate, the more load you put on the muscle and the more tension it has to generate, the slower the velocity of contraction, because the muscle is contracting against a higher and higher load, and it's slowing that contraction down. And so my velocity of shortening decreases as the load on the muscle increases, until finally I get to this point. And this is a point where I put so much load on the muscle that it's no longer allowed to shorten at all. So now I'm stimulating this muscle fiber. It can't shorten at all because I put such a load on it, I'm holding it in place and not allowing it to shorten. This is what's called isometric contraction. It generates maximum tension with no change in length and therefore no velocity of contraction, okay? And that's where you're simply holding the muscle such that it can't shorten. You've put a maximal load on it. And so the muscle generates as much tension as it possibly can in an effort to shorten it, it can't. So it generates maximum tension at that point with zero velocity of shortening. So how does contraction of a single fiber vary? First of all, it'll change with the length. I think we just went through that, right? So this is the contraction of a single fiber. This is an increase in force here over time, and I've stimulated it twice. And if I change the fiber length, for instance, if I lengthen it beyond optimal, then this twitch force will go down, okay? I can stimulate really quickly so that I stimulate once, and then before the muscle has a chance to relax, I stimulate again, and what I get is an addition to the first stimulation. So I get summation, and you can do that. In your body, you can cause action potentials to fire repeatedly, and you can cause the amount of tension generated to build because you don't allow the muscle to relax. And of course, if you do this to the maximum, then you get what's called tetanus. This is tetanus here. This label should be down here, and it should be on your slide at home. Down here, this is where you've stimulated it so many times that you've achieved maximal tension and not allowed that muscle to relax at all. This is called tetanus. Now you'll recall cardiac muscle, you can't tetanize. And the reason is because you always get a significant amount of relaxation before you can stimulate that cardiac muscle again. That's because the refractory period of the action potential is much longer in cardiac muscle. You have this long plateau phase where calcium's coming in, and during that plateau phase, you can't re-stimulate the fiber. And you can't re-stimulate it until it gets almost all the way down to rest. So in this case, because the action potential is so much shorter in skeletal muscle, in skeletal muscle, you can repeatedly stimulate this really quickly over and over again and never allow it to relax. You can't do that in cardiac muscle. Cardiac muscle always relaxes to a certain extent before you can re-stimulate it because it remains refractory for too long. And I think that's something that you want. If your cardiac muscle never relaxes, your heart never fills, because you never get diastole, and that would not be good. Just to have a hypercontracted heart that never relaxes, that means that your cardiac output falls to zero, and that's not great. So you can think of that as a safety factor where the muscle simply won't allow the action potential to re-stimulate it again until it's had a certain amount of time to relax and fill a little bit. Okay, Single fibers can also, the force can change with changes in fiber diameter, bigger force. With fiber type, which we'll go over in a second, type 2 tend to generate more force than type 1. And with fatigue, if you repeatedly stimulate these muscle cells, they fatigue to the point where they won't contract very well. They get tired, okay? And so if they get tired, they don't generate as much force. Your body tends to alter the force of contraction by a process that we call recruitment. So you have a motor unit, which is a group of fibers which is innervated by one neuron. So you have one neuron coming down, and it's causing neuromuscular junctions with multiple fibers, and all those multiple fibers, any fibers that are innervated by one neuron, that's called a motor motor unit, because those fibers all contract together in response to stimulation by the nerve. Those do vary in size. They tend to be bigger in muscles that have to generate a lot of force, where the movement isn't really fine. You're not really talking about fine regulation of movement. Big muscles, that generate a lot of force without a lot of fine contraction, those tend to have big motor units. Muscles that need really fine control, like your eyes, or maybe the muscles in your fingertips, where this movement has to be really fine and well controlled, those tend to have smaller motor units. So what your body does is it recruits more motor units when it needs to generate more force in a muscle, okay? And unless you're actually recruiting as many fibers as you possibly can and generating maximal contraction, that process is asynchronous. So you get asynchronous recruitment, and this is especially in postural muscles. What happens is your body's pretty smart. It won't just repeatedly stimulate the same motor unit if it's just trying to generate a submaximal contraction, because that motor unit will eventually fatigue. So what it does is it stimulates one motor unit, and then it allows that to relax while it stimulates another motor unit and recruits it to maintain tension. And then that one can relax while it stimulates another motor unit to maintain tension. So you maintain a constant tension in the muscle by asynchronously recruiting these one at a time. So one motor unit doesn't bear all of the weight and all the fatigue, okay? That is another form of recruitment. So in the case of multiple fibers, you recruit motor units until you've generated enough force. And if it's a sub-maximal amount of force that you've had to generate, then that recruitment becomes asynchronous, where you're alternating stimulation of motor units, because you don't need them all to be stimulated all at once, okay? And finally, in order to go over fiber types, we have to very quickly review metabolism and skeletal muscle fibers. I'm sure that Dr. Szabo has gone over this in enormous detail with you, and I am certainly not going to do that. I don't know it in enormous detail. But I know enough of it to be able to describe the muscle fiber types, which I'm going to get to on the next slide. So let's talk about this a little bit. Here's your muscle fiber. Of course, you need ATP to contract. However, there's only so much ATP, and that ATP is depleted fairly quickly in a fiber when you stimulate it to contract. So the first thing you do is you start to use creatine phosphate in the fiber. There it is, right there. Creatine phosphate here, which will hydrolyze and transfer an ATP to phosphate, an energized phosphate, to ADP to form ATP. So you form more ATP from creatine phosphate, and that maintains your ATP, at least for a little while, in the face of contraction, okay? Of course, that depletes fairly quickly as well, and so your muscle does have to be able to produce energy, ATP, through metabolism of glucose and other metabolic substances, and it does that through oxidative phosphorylation and glycolysis. So oxidative phosphorylation you have here, very efficient, and that it generates a lot of ATP per glucose molecule, per molecule of food energy. However, it is slow and it requires oxygen, so it's not ideal for all circumstances, okay? On the other hand, you have glycolysis, which is less efficient in that it does not generate as much ATP per glucose, but it's fast and it does not require oxygen, and so that makes it ideal for certain situations. And there's our glycolysis right there. Okay, so all muscle fibers use both forms of metabolism. However, they do use them to very different extents. And that is part of the basis of separating your muscle fibers, because muscle fiber types, some fiber types tend to use glycolysis, some tend to use oxidative phosphorylation. Okay, just to finish this up, we have three kinds of fiber types here. They can be split into two groups. The type 1, which are actually unstained on this micrograph. That's because the stain is for myosin ATPase activity. These have low myosin ATPase activity. The type 2 have a high myosin ATPase activity. Therefore, they're darkly stained here. And you can see that this muscle is a mixture of those two types. The type 2 generally have a higher SR calcium pump activity. They have more parvalbumin. And so what that does is it shortens the contraction duration, but as we'll see in a minute, they tend to generate more force. So the SR calcium pump takes up the calcium quicker. The presence of the parvalbumin binds the calcium and brings it back down after the peak of the calcium transient, and that shortens the duration. But again, we have a higher force of contraction in these muscles, largely due to the higher myosin ATPase activity, of course. And again, most muscle contains a mixture of these fiber types, but the amount of each that you have varies substantially, and often you will hear muscles classified as type 1 or type 2, depending on the situation, or mixed. So first of all, let's start with the type 1 fibers. Again, low myosin ATPase activity. They tend to be smaller in diameter, and they depend on oxidative phosphorylation for most of their energy, and therefore they need plenty of oxygen, and therefore, not surprisingly, you have a high density of capillaries in these muscles. And you tend to have a high density of mitochondria, of course, because they're utilizing the oxygen to generate your ATP. You also have myoglobin in these fibers. They're typically called red fibers because of the presence of the myoglobin. The type 2 are typically white fibers. And they have a lower glycogen content because they're more efficient at turning that the glucose that they get into energy, so they don't need quite as much. They are relatively small motor units, and perhaps most importantly, they are highly resistant to fatigue. So this is tension generated here, and this is time on the x-axis. And you can see, as we repeatedly stimulate this muscle fiber, it continues to generate the same tension on, and this is 60 minutes here, and it's still generating the same tension, so it's not really fatiguing very much. On the other hand, we have type 2B fibers. These are fast glycolytic fibers, so not slow oxidative, fast glycolytic, so they are the other end of the spectrum. They have a low capacity for oxidative phosphorylation, and therefore they are very dependent upon glycolysis and anaerobic generation of ATP. They fatigue rapidly, so you can see, as opposed to the type 1, which go on and on like the Energizer Bunny, the type 2, the tension falls over time pretty rapidly here, the type 2B. They have few mitochondria. They don't need very much. They have very little myoglobins. They're called white fibers. Again, very powerful but very brief contractures in these muscle fibers. And they have a large store of glycogen because they're not as efficient at utilizing glucose. They need a lot of glucose around in the form of glycogen in order to sustain what activity they can. Okay? So our last fiber type is sort of in between these two. They're the type 2A. They're fast, oxidative fibers. Okay? So they have a high capacity for oxidative phosphorylation. Okay? Not as dependent upon glycolysis as the type 2B. They are nevertheless, they have very big forceful contractions that are much more like the type 2 than type 2B than the type 1. They have an intermediate diameter, an intermediate motor unit size, intermediate capillary supply, intermediate glycolytic capacity, better than this, better than this, not as reliant as this, less reliant, more reliant on glycolytic capacity. They have an intermediate glycogen content and intermediate myoglobin content. They are considered to be white fibers. And they are resistant to fatigue, more resistant than the fast glycolytic, which really go down fast, but not as resistant as the type 1 slow oxidative, which go on and on and on. These generate a goodly amount of tension for a good period of time, but see how they fall off. They don't fall off as quickly as the type 2B, the fast glycolytic, but they do fall off more rapidly than the type 1, the slow oxidative. So the fast oxidative fibers are intermediate, okay? So that's all I've got. Thank you for your attention, and I'll see you guys in the sessions.